thank you so much to the organizers for this beautiful event. I'm triple lucky today because I'm not on the organizer committee, so it's very smooth sailing, no stress, and I get to present my work to this wonderful audience here on home turf in Maastricht. And also I follow this beautiful keynote and other talks on predictive processing, so I feel like I have a very easy job, and I'm very lucky to have this opportunity. I will talk about auditory predictions, how they relate to memory, and how task context may shape predictive processing. What I mean by predictive processing is a relatively abstract or conceptual framework that posits that the brain entails a generative model of its environment. This model consists of predictions that are propagated in a top-down manner from higher to lower brain regions, and at each stage they are compared with the incoming inputs, either from the sensory periphery or from lower order brain regions. And the result of this comparison is a prediction error that is then propagated back up the hierarchy and is used as a teaching signal, so it is used to update this model. Now, this relatively abstract framework has been proposed to or translated to a specific mathematical formalism or an algorithm called predictive coding. And predictive coding comes in several flavors or versions, but one of the more popular ones posits that prediction and prediction error units are separable, that they express or encode different quantities, for example, the causes of sensations versus more dynamic trajectories of the neural states. Perhaps more interestingly for us, that the prediction errors are modulated by precision, such that in most cases a prediction error will be used to update the prior, but very precise prediction errors will have a stronger influence on this updating process. And this algorithm has then been proposed to be implemented in a particular neural mechanism. And this neural mechanism, for example, could entail some spectral asymmetry between prediction error and prediction signaling. So, for example, prediction errors have been proposed to be signaled in higher frequency bands than in predictions. Perhaps they are signaled by more superficial cortical layers than predictions. And this precision modulation term has been linked to a type of neural gain control, for example, implemented by neuromodulation. So this has been proposed as a sort of a canonical mechanism for predictive coding. And what our work has focused on in the last years is to test the universality of this supposedly canonical mechanism. So in other words, we are testing to what extent different task contexts, different types of predictions actually are implemented with this canonical mechanism or with different types of mechanisms. And let's start with a relatively rudimentary form of predictive processing, and it also has been introduced beautifully yesterday by Bernhard Englitz. So here we analyzed data collected by Vani Rajendran and Nicole Harper in Oxford. These were direct recordings from the primary auditory cortex of anesthetized rats. And these rats were exposed to very simple sequences of noise bursts. These noise bursts are here represented by these vertical dashed lines, and they were regularly presented at these isochronous rates. Now, occasionally the noise bursts were omitted, and this is shown as these little red dots over here. And even in these example traces, I think you can see that there were very clear omission responses. So these are responses to sounds that would be expected to be played but were omitted. When we analyzed multiple trials, when we averaged over trials, we saw clear evidence for these omission responses. So here in solid lines, you can see sound-evoked responses locked to the sound onset, and in the dashed lines, you can see omission responses. So these are now locked to the onset of an expected sound that isn't actually there. And these are temporally specific. So we see this nice time-locked response, omission response, at different presentation rates, so suggesting that we have a very temporally specific omission response to the omitted sound. These were present both in lower and in higher frequency bands, so in the LFP range and in the AMUA range. And when we looked at different layers, we actually found that while both deeper and superficial layers had some omission responses, in the superficial layers there was a relatively strong contribution of these faster frequency bands, so in the AMUA range. So this suggests that even in these anesthetized rodents, perhaps there is some evidence for both a laminar and a spectral specific contribution to predictive processing. But of course, regular noise bursts are not usually how we predict our environment, right? So for example, in speech, we typically extract a little bit of the stimulus history to predict what the person will say next, and same goes for music. And again, we are not the only species who do that, and at this meeting there were also some beautiful presentations and contributions about the evolutionary conservation of some of these mechanisms, for example, in music processing or in artificial sequence processing between, for example, humans and non-human primates. What we wanted to do was to test this rodent model a bit further. And so we typically then, when we want to study predictive sequence processing, we use these kinds of more controlled sequences, as for example in statistical learning, where different elements occur over and over again in a continuous stream, but some of them co-occur more frequently than others. So when you are exposed to such a stream, after some time, usually you would implicitly learn that, for example, this element P is more likely to be preceded by the element 2 than by another element Go, for example. And to process this sequence in a predictive manner, you not only have to have a memory of the immediately preceding element, because this means that you are now processing, let's say, a particular regular triplet in this case, but you also need to have a slightly longer term memory that this triplet has reoccurred in the past. And you usually form a prediction, and if this prediction is violated, you will probably have a mismatch response, as we've heard this morning. So in these next couple of studies, we tried to actually dissociate or try to access the contents of mnemonic and predictive processing in the auditory cortex. And to do that, we first wanted to establish a cross-species model of decoding memory, because this was much more established in the human literature, and we wanted to translate it to the rodent literature. And then, once we've established that, we want to test it on also decoding auditory predictions. So in this first cross-species study, we had human EEG data in humans performing a task, and rodent or rat electrophysiology data. So here, these were electrocorticographic recordings from the surface of both primary and secondary auditory cortex. The human participants were asked to perform a very simple delayed match to sample task. So there was an initial tone with a particular frequency and a later probe tone with another frequency, and the participants were asked to judge which of these frequencies was higher or lower. But importantly, in between these two tones, so in the sensory memory retention interval, we presented this impulse, which is a noise burst, but it's meant here to potentially reactivate a silent memory trace or be used to read out the contents or decode the frequency of the tone held in sensory memory during the retention period. In the rodents, these were anesthetized rats, so we didn't implement any task, but we simply interleaved these pure tones with the impulses. And again, we want to decode the frequency of the preceding tone from this impulse-evoked response. And when we did that, on the left you can see the decoding time series locked to the sound onset, so to the pure tone onset, and here we can decode very well the tone frequency from the tone evoked activity, both in humans and rats. But on the right you can also see that we can decode the frequency of the preceding tone from this burst or impulse evoked activity, again both in humans and rats. So here we establish a decoding method that can generalize across species and data modalities. And we then use this method to access a slightly more predictive signal also in a more complex sequence. So here we use these sort of triplets. In this case, we actually used artificial vowels. And so these triplets of vowels repeated over and over again, and then maybe they would be replaced by another triplet, and again, this would repeat over and over again. Occasionally, we replaced some of the triplet elements with these impulses or with these noise bursts. And here the logic was that if we can decode the immediately preceding tone, this is tapping into memory processing, so sort of replicating the previous study. But since these are triplets, what we should also be able to decode is the identity of the expected vowel at the time when we are presenting these impulses. And this should only be possible in the predictable blocks when these triplets repeat, but not in the random blocks where the sequence is shuffled. So indeed, on the left, you can see our replication of the memory study. So here we find significant decoding of the immediately preceding vowel identity that is very similar for both predictable and random blocks. So there was no significant difference between these two conditions. Again, just tapping into sensory memory. But on the right, you can see that in the predictable blocks, we could additionally decode the identity of this expected vowel that is replaced by the impulse, and this was not the case in the random blocks. So here we are tapping into an additional decoding of the predictive component of these sequences. So to summarize these studies so far, it seems that some aspects of predictive processing may be automatic in the sense that they can be observed in the rodent models even under anesthesia. These are completely naive rats, never exposed to these sequences before. So it's quite striking that we can still decode these predictive sequences in those rats. But then we thought, well, maybe let's test it even further. And so here we actually played these kinds of interleaved triplets of artificial syllables to rats. But prior to the recording under anesthesia, some of these rats were actually actively trained, so they were instrumentally conditioned to discriminate specific triplets from other shuffled triplets. Other rats were simply passively exposed for overnight before the students came in the morning and started the recordings, and some rats were naive. And here we found that, first of all, for the initial syllable, there was no difference between familiar and unfamiliar triplets because these are actually physically the same in the familiar and unfamiliar sequences. So you need to process the later elements to know that you're now processing a familiar triplet. And so at the last element of a triplet, we found significant boost in decoding for familiar versus unfamiliar triplets, but only for those rats that were actually actively trained before the recording. So it seems that we're here hitting a limit of how useful these anesthetized rodent models may be. So you really need to very extensively train these animals to then tap into processing much more complex interleaved sequences in these animals. But of course, in humans, the sky's the limit when it comes to overcomplicating paradigms, right? So here we decided to also analyze human data. So here we implemented a task in which we tried to look at the interactions between predictability and contextual relevance based on what is relevant for you in a particular task. So if you hear a loud sound, right, like a car honking, this might mean that there's a vehicle traveling very fast towards you. And so in those contexts, your spatial and temporal predictions of where that car might be are very relevant. But in other contexts, in a traffic jam, a car honking might just mean that someone is frustrated or bored, right? So in that case, it really doesn't matter where or when they will be in a second. So we, in this study, wanted to test to what extent contextual relevance interacts with predictions, but also if these task effects can modulate the neural signatures of either predictions or prediction errors or both. And we designed this cross-model audiovisual probabilistic cueing task in which there was an auditory cue and a visual target. The pitch of the cue predicted the location of the target, and the composition of the cue, so whether it's an ascending or a descending pair of tones, predicted the latency of the target. And this was completely unknown to the participants. So participants were not informed of these contingencies. They were also not informed that these contingencies keep changing over the course of the experiment. So for example, you might start with several trials in which, for example, high pitch predicts left targets with 90% probability, but then maybe you switch to a 50% probability and then to the other type of prediction where high pitch now predicts right targets and so on. So this keeps evolving. These two contingencies were uncorrelated, so they were completely independent from each other. And the only thing that we told our participants is what is relevant for them in a particular block of the trials. So they were asked to either perform a spatial discrimination task, categorize left versus right, or a temporal discrimination task. When we first looked at behavior, we saw already clear evidence for this interaction between contextual relevance and predictability. So we found that spatial predictability only increased accuracy in the relevant spatial task, but not in the irrelevant temporal task, and vice versa for the temporal predictability. But we wanted to go deeper into understanding this behavioral result. And so for this purpose, we turned to computational modeling. So in our lab, quite often, we use different computational techniques to bridge this gap between the theories that we try to test and the data that we have access to. And so I already showed you an example of some decoding techniques, but now we can also work in a more top-down manner where we actually start with a theory, what we think the person might be doing, and then we estimate some trial by trial, for example, predictions that the person might have, and then we use it to correlate it with neural data. So in this case, the model that we used is a so-called hierarchical Gaussian filter, which is a model of belief updating, a trial-by-trial model of learning. And in this case, the model is a generative model of behavioral responses, button presses in this task, and it's fitted to individual person's data. But it also assumes that the person has some predictions about what is happening in the task. And these predictions are hierarchical in the sense that at the lowest level, level 1, these are predictions about a particular target being, for example, on the left or on the right, or early or late in a particular trial. At the intermediate level, these are predictions about the contingency between the cue and the target. So is it now high pitch predicting left or right? And at the highest level, these are predictions about the volatility of this task. So are things changing very dynamically, or are they very stable over trials? And what we get out of this model when we fit it to individual behavior are these time series of trial-by-trial estimates of that person's predictions and prediction errors, separate for the spatial and temporal predictions. And what you can see here is just an example of one participant fit, where the colored lines are the model estimates of these probabilities, and the gray lines are the actual objective levels of predictability, which were, again, completely unknown to the participants. So the model fits these experimental manipulations very well. And now when we tried to link this to neural data, in this case we had MEG recordings, and we looked at time frequency activity, so at induced neural oscillations in response to these cues and targets. And we found nothing. And this was, of course, very concerning to us because, well, we have this clear behavioral result, so we would expect some sensitivity to predictability in our neural data. But when we then looked at the difference between relevant and irrelevant predictions or prediction errors, we indeed found very clear dissociation in the different frequency bands, such that relevant predictions modulated lower frequency bands in the auditory regions because these were the cue processing regions, and relevant prediction errors modulated higher frequency bands in the upper beta and lower gamma bands in the visual regions and also in sort of associative regions in the TPJ. So here we have evidence for not only a dissociation between prediction errors and predictions in terms of which frequency bands they are signaled by, but also that contextual relevance very strongly modulates these neural signatures of both predictive processes. So in more complex cases, like these interleaved sequences in the rodents or these very complex probabilistic paradigms in humans, the task actually does affect predictive processing in a way that it very strongly modulates the neural responses to these predictive sequences. But task relevance is not the only context, and time is another very important context. The same stimulus may be very different and have very different effects on your surprise, whether it's temporally predictable or not. So in these next couple of studies, we tried to dissociate what predictions for stimulus contents from when predictions for stimulus timing. And here again, we had a cross-modal design, but this time the cues were visual and the targets were auditory. So in general, this face of a visual image could predict in some blocks the identity of the syllable that was played to these participants. For example, Obama in the predictable blocks could predict pa with 75% probability, and your other favorite president could predict ga with 75% probability. So we had these predictable blocks. We also had unpredictable blocks in which these were 50-50, so there was no relationship between the face and the sound. And orthogonally to that, we also had temporal predictability manipulation. So in some blocks, these stimuli were presented at fixed regular intervals, and in other blocks at random irregular intervals. And in this case, we first analyzed ICAC data. So these were, again, recordings from the cortical surface of patients undergoing, in this case, pre-surgical monitoring for pharmacologically intractable epilepsy. And when we looked at, first of all, the validity effects, so these are the differences between mispredicted sounds and correctly predicted sounds, so sort of a prediction error response. This was a very widespread modulation of activity basically throughout the temporal and frontal regions. But when we looked at this contextual predictability manipulation, it was more specific. So what predictability mostly affected activity in the frontal regions, when predictability also affected activity in the sensory motor regions, so there was an additive effect of both kinds of contextual predictability, and they both interacted in the superior temporal gyrus. And we were also interested in diving deeper into these results at the surface level. So in the first instance, again, we turned to computational modeling, in this case, dynamic causal modeling. So this is a model of connectivity in which you build different connection structures between different brain regions, but also these connections might be intrinsic, so they are between different populations within brain regions. And then you compare alternative hypotheses of how this connectivity might mediate different types of experimental manipulations. And then usually you find your winning model, or you average over many models. And in our case, this is already published, we found that these two predictability manipulations had very different connectivity effects. So not only between regions, but also within regions. We implemented two different types of gain control in these models. One type of gain control was top-down, so the neural gain of the STG depended on top-down inputs from other parts of the network. And another type of gain modulation was local, so it was independent from what was happening in other parts of the network. And we found that what predictability mapped onto this more top-down dependent gain, whereas when predictability modulated local gain. So we interpret this in terms of putative differences in neuromodulation. So for example, the top-down gain could be more driven by something like NMDA-dependent short-term plasticity, and the local gain could be due to classical neuromodulation or perhaps entrainment to these rhythmic structures. But more recently, we also were very lucky to analyze data recorded from a subset of these patients, but these are now laminar data. So these are laminar probes implanted just under the ECOG grid that span the entire depth of the cortical sheath in the STG. These are pretty rare recordings in humans. Only a handful of labs in the world perform these recordings, and here we analyze patients from New York and from Budapest. And when we looked at the laminar profiles of these effects of different types of predictions or contextual predictability, again, we found dissociations between them. So first, if we look at, again, the validity effect, so putative prediction error, we find mostly modulations of superficial activity. But when we look at these more contextual effects, this is also expressed in the deeper layers. And for example, temporal predictions only affect the deep layers in these data. And they also interact in the input layer, so in layer 4, which could, again, be consistent with this idea that it sets the gain to incoming inputs into layer 4 in the STG. Now, coming back to decoding, we could also decode the syllable identity very well in the superficial and deep layers following syllable onset, so this is maybe not as surprising. But we could also decode the predicted syllable independent of what will actually be presented to those participants. And this decoding was significant only in the deep layers before syllable onset. So here we have relatively clear evidence that predictions in terms of actual decoding of what is being predicted are expressed mostly in deeper layers in these neocortical regions. And in very recent work, Mehdi Enan, and also this is supervised, it's a collaboration with Federico De Martino, they translated this paradigm into a 70 fMRI study, and here I'm not showing you the entire result because Mehdi also presented this as a poster a couple of days ago, but in the hippocampus we could segment the hippocampus into different subfields, and here we found this decoding of the predicted syllable specifically in the CA23 subfield of the hippocampus. So I'm not arguing that everything is happening in the neocortex. Actually, the hippocampus is also most likely a very important hub in processing these types of predictive sequences. Now, what I'm not showing you today, but I would just like to briefly mention it, and this is my last slide before I conclude, is another line of work that I find quite exciting, and this is about how predictions relate to learning. So I mentioned that prediction error is a type of a teaching signal, so it's used to update the model. But actually, this suppression of this effect of prediction on ongoing activity could be implemented in two different types of mechanisms, a dampening mechanism and a sharpening mechanism. And there's this long literature on these dissociations between different mechanisms of predictive suppression. And what we did with my PhD student, Hannah McDermott, is that we looked at this more systematically in a learning paradigm where people actually did not have any repetition of any stimuli because everything was unique. And I'm also not showing this to you because I thought it would be a bit blasphemous to show a visual study for an auditory cortex conference. But luckily, Hannah has also translated this to a cross-modal paradigm. So now she has an auditory component, an auditory cue in these paradigms, and she also tested this at 70 fMRI. So if you're curious about these results, please see her at her poster later today. So to conclude, we find that some aspects of predictive processing may well be automatic. In more complex cases, they are very strongly modulated by context, and not all predictions are equal. So for example, predictions for stimulus content versus timing may actually rely on very different brain circuits, even at the laminar level in the neocortex. And with that, I would like to thank you for your attention, and thanks to my collaborators and sponsors. Thank you.
First. Thanks a lot, Ryszard, for this beautiful overview of your work. I was curious, because jumping back to the very beginning of your talk, and you being one of the few people that has peered deeply enough into Carl Friston's mind, is the division between the cells, prediction errors, is that something that you would consider necessary for this organization, or do you think that new theory by Flo and Mikuláš is equivalent and could be seen as implementing the same principle, just but with a different anatomical, or physiological integration?
Yeah, great question. And actually in that omission study, for example, we also, what I didn't show you is that we didn't find evidence for actual spiking activity underlying these omission responses. So we did interpret it tentatively that perhaps it could be due to dendritic predictive processing, and I think this is now quite an active field of research. What I could also perhaps draw your attention to is this big collaboration orchestrated by the Allen Institute, where they look at different types of mismatch sequences, so oddball sequences, sensory motor mismatches, omissions, and they actually try to adjudicate between these hypotheses, so between sort of a classical predictive coding with different units and more dendritic predictive coding with different compartments. And I think that's a great open question out of the field, to what extent we can actually translate these schemes into a subscale of dendritic predictive coding.
Yeah, over there.
Thanks, Ryszard. The hippocampal slide you showed at the end, that was fascinating. Were the contents that were being predicted there, did you say they were purely simple auditory sounds, or were they more complex?
They were syllables.
Okay.
So they were Pa versus Ga. In that study, we also had omissions in the 70 fMRI study, but yeah, so this is just a prediction of a syllable, one or another.
And would you have some hippocampal coverage in your intracranial data as well, presumably? Are you looking at that as well?
There was some coverage, but not in the same patients that we had the laminar recordings. Okay. So that is a bit tricky to integrate. But there was there were some patients, of course, with hippocampal coverage, and I don't think the person who was working on those data, this is Ji-Yoon Shin at New York University, I don't think she looked at decoding. But what she did find that was different from the cortex is that in our hands, all of these predictive signals at the level of LFP amplitudes or univariate results, they all happen after the syllable onset, but in the hippocampus they actually happened before syllable onset. So there was already some ramping activity in the hippocampus in the predictable conditions there.
Thank you.
Thanks, Richard, for this extremely interesting talk. I had a question about the what and when study. In the end, you mentioned the decoding results, that you could decode the predicted syllable. Was the decoding performance somehow specific in time, and if so, like related to the temporal predictions? Like, could you decode it in the time when it was predicted to appear, or did you look at it?
There were no differences between these two conditions. So yeah, this was pooled over them, and then when I tested for differences, I didn't see any differences between these two conditions. Yeah, but great question, thank you.
Thanks.
Yeah.
Question about comparing your results to Vicario's results in the anesthetized birds, where I just was reminded by the segmentation of words and all that stuff, which also shows quite strong effects of predicting elements within the words and all that. And I was just curious whether you think it makes any difference which species, whether you're using rodents versus birds, and whether his results are give you different conclusions as to the limits of anesthetics and so on.
Yeah, thank you. Well, yeah, I can only speak from these data that we have basically evidence for this relatively complex memory and prediction signal in some sequences, but then not in much more complex sequences. But of course, there could be some species differences. So especially in species that do use sequences for much more complex processing, where you have these different chunks and at different levels, they are also related to each other. It could be differences there, right? But yeah, I don't have data to speak about that. Thank you.
Okay, if there are no further urgent, Elia, yeah?
Hi, thanks a lot. In the initial theoretical slide, right, you mentioned that these predictions are very precise at the sensory level even, right? I mean, high spatiotemporal precision or something like this. But I mean, in many of the presentations during this conference, all these predictions, for example, are not tonotopic, right? So all the effects are not tonotopic. How do you reconcile this?
Well, I think there is very nice work, for example, from your neighbor, from Federico, showing some tonotopy of, for example, omission responses, right?
The one of Werner, not.
Yeah, correct. So in our case, we don't, well, we rarely look at tonotopy. Yeah, and in these cases, we also don't do that. There is one study that we did, but this is based on population receptive field estimation based on MEG and EEG, right? So, of course, there's only a certain amount of tonotopy that you could infer from that. And in that case, for example, we also didn't find evidence for very tonotopic modulations or spectrally specific modulations with respect to this temporal prediction. So yeah, it could be different depending on what type of prediction you're talking about. For example, temporal predictions in our hands are also very generic, so they don't specifically augment those parts of the population receptive field that are tuned to what you would be expecting in terms of contents. Yeah. Thank you.
Okay, I